Welcome to Chapter 7, Hypothesis Testing. This video content is based on Essentials of Biostatistics in Public Health by Lisa M. Sullivan. Public health research often asks questions like, does this intervention work? Is this new treatment better? Are these two groups different? To answer these questions, we use hypothesis testing, a statistical process that helps us decide whether the data supports a specific claim. In this chapter, we'll walk through hypothesis testing for a variety of outcome types, using sample data to make inferences about populations. First is Introduction to Hypothesis Testing. Hypothesis testing starts with two competing ideas. The null hypothesis, or H0, assumes there is no difference or no effect. The alternative hypothesis, or H1, suggests there is a difference or an effect. We also define a significance level, often 5%, which we call alpha. This is the threshold for deciding whether to reject the null hypothesis. A type 1 error occurs when we reject the null hypothesis even though it's actually true, a false positive. A type 2 error happens when we fail to reject the null when the alternative is actually true, a false negative. The p-value tells us how likely we are to see our results, or more extreme if the null hypothesis is true. If the p-value is less than alpha, we typically reject the null. Hypothesis testing is closely related to confidence intervals. If the CI doesn't include the null value, the result is statistically significant. Second is test with one sample. Continuous outcome. Suppose we want to know whether the average sodium intake in our sample is different from the recommended value. We use a one-sample t-test. We calculate the test statistic by subtracting the hypothesized mean from the sample mean. Then we divide by the standard error of the mean. Take the sample mean minus the hypothesized value, then divide by the standard error. If the resulting t-value is large in absolute value and the p-value is less than alpha, we reject the null. Third is test with one sample, dichotomous outcome. What if we want to test whether the vaccination rate in our sample is different from a goal of 80%? We use a z-test for proportions. Subtract the hypothesized proportion from the sample proportion, then divide by the square root of the hypothesized proportion times 1 minus the hypothesized proportion divided by the sample size. We then compare the resulting z-score to standard normal distribution values. If the p-value is below alpha, we reject the null. Fourth is tests with one sample, categorical and ordinal outcomes. When we want to compare observed counts to expected counts in a single group, we use a chi-square goodness of fit test. Example, do observed blood type frequencies differ from what we'd expect? We compare each observed count to its expected count. For each category, subtract the expected from the observed, square the result, then divide by the expected count. Sum those values across all categories to get the chi-square statistic. We then compare that value to a chi-square distribution with the appropriate degrees of freedom. Fifth is tests with two independent samples, continuous outcome. To compare the means of two unrelated groups like males versus females, we use the independent samples t-test. Subtract the two sample means, then divide by the square root of the sum of each group's variance divided by its sample size. If the result is significant, we conclude the means are different. If the assumption of equal variance isn't met, we apply a version called Welch's t-test. Sixth is tests with matched samples, continuous outcome. When the same individuals are measured twice, like before and after an intervention, we use a paired t-test. Take the average of the differences between paired observations. Then divide by the standard deviation of those differences over the square root of the sample size. If the p-value is low, the before and after results are significantly different. Seventh is tests with two independent samples, dichotomous outcome. Suppose we compare smoking rates between two unrelated groups. We use a chi-square test for independence or a z-test for two proportions. We calculate a test statistic based on the difference in proportions. If the p-value is low, we conclude the proportions are significantly different. Eighth is tests with more than two independent samples, continuous outcome. When comparing means across more than two groups, say 3 different diets, we use analysis of variance or ANOVA. ANOVA tests whether at least one group mean differs. It compares the variation between groups to the variation within groups. If the p-value is below alpha, we conclude not all group means are equal. Post hoc tests are then used to identify which specific groups differ. Ninth is tests for two or more independent samples, categorical and ordinal outcomes. To compare distributions across categories or groups, we use a chi-square test for independence in contingency tables. For each cell in the table, subtract the expected from the observed count, square it, divide by the expected, and sum across all cells. Used when analyzing preferences, behaviors, or exposures across regions or demographics. Conclusion hypothesis testing helps us move from observation to decision. In this chapter, we covered tests for one sample and two samples, continuous, categorical, and ordinal outcomes, matched samples, multiple group comparisons. We also learned how p-values, test statistics, and assumptions guide our interpretation. Hypothesis testing is a cornerstone of biostatistics and public health evidence. Next, we explore correlation and regression to describe and predict relationships between variables. This video content is based on Essentials of Biostatistics in Public Health by Lisa M. Sullivan.